डियर फ्रेंड्स, आज हम डिस्कस करेंगे एनाटॉमी एंड फिजियोलॉजी ऑफ सेल्स। Cells are the structural units of all living things. All cells arise from existing cells by the process of cell division, in which one cell divides into two identical cells. The English scientist Robert Hooke first observed plant cells with a crude microscope in the late 1600s. Then in the 1830s, two German scientists Matthias Selden and Theodor Schwann proposed that all living things are composed of cells. Cell diversity. Parts of cell. Human cell has three main parts: the plasma membrane, the outer boundaries of the cell. सेल की जो आउटर बाउंड्रीज होती है उसको प्लाज्मा मेम्ब्रेन कहा जाता है, एंड द साइटोप्लाज्म, द इंट्रासेल्युलर फ्लूइड्स packed with organelles, small structures that perform scientific specific cell functions. The nucleus and organelles that control cellular activities. The nucleus lies near the cell center. The plasma membrane defines the extent of a cell, thereby separating two of the body's major fluid compartments: the intracellular fluid within the cells and the extracellular fluid outside the cell. The plasma membrane encloses cells, contains, mediates exchanges with the extracellular environment, and plays a role in cellular communication. Structure. The fluid mosaic model. The fluid mosaic model of membrane structure depicts the plasma membrane as an exceedingly thin 7 to 10 nanometer structure composed of a double layer of lipid molecules with protein molecules dispersed in it. The protein which float in the fluid lipid bilayer form a constantly changing mosaic pattern. The glycocalyx. Plasma membrane contains the carbohydrate-rich area at the cell surface called glycocalyx. Cell function. Cell junctions. An epithelial cell is joined to adjacent cells by the three common types of cell junctions: tight junctions, desmosomes, and third last is gap junctions. Tight junctions, impermeable junctions, prevent molecules from passing through the intercellular space. Desmosomes, anchoring junctions, bind adjacent cell together like a molecules velcro and help form an internal tensor reducing network of fibers. Gap junctions, communicating junctions, allow ions and small molecules to pass for intercellular communication. The cytoplasm. The cellular material between the plasma membrane and the nucleus is the site of most cellular activity. It consists of three elements. First is the cytosol, and second one is organelles, and last one is inclusions. The cytosol, it is a viscous semi-transparent fluid in which the other cytoplasmic elements are suspended. The organelles are the metabolic machinery of the cell. Each type of organelles carries out a specific functions for the cell. The inclusions are chemical substances that may or may not be present depending on cell type. Mitochondria. Red-like double membrane structures. Inner membrane folded into projections called cristae. Site of adenine tryptophosphate synthesis, powerhouse of the cell. Ribosome. Dense particles consisting of two subunits, each composed of ribosomal RNA and protein, free or attached to rough endoplasmic reticulum, the sites of protein synthesis. Rough endoplasmic reticulum, membranous system enclosing a cavity, the cistern and coiling through the cytoplasm, externally studded with ribosomes. Sugar groups are attached to proteins within the cisterns. Proteins are bounded in vesicles for transport to the Golgi apparatus and other cells. External phase synthesized phospholipids. Smooth endoplasmic reticulum, membranous system of sac and tubules free of ribosomes, site of lipid and steroids, cholesterol synthesis, lipid metabolism and drug detoxification. Golgi apparatus, a stack of flattened membranes and associated vesicles close to the nucleus. Packages, modifies and segregates proteins for secretions from the cell, inclusions in lysosomes and incorporation into the plasma membranes. Peroxisomes, membranous sac of catalase and oxidase enzymes. The enzymes detoxify a number of toxic substances. The most important enzyme, catalase, break down hydrogen peroxide. Lysosomes, membranous sac containing acid hydrolases, site of intercellular digestion. Microtubules, cylindrical structures made of tubulin proteins, support the cell and give it shape, involved in intracellular and cellular movement from centrioles and cilia and flagella if present. Microfilaments, fine filaments composed of the protein actin, involved in muscle contraction and other types of intracellular movement, help from the cell cytoskeleton. This is actin subunits and this is 7 nanometers. Intermediate filaments, protein fibers composition varies. The stable cytoskeleton elements resist mechanical forces acting on the cells. This is 10 nanometer. These are intermediate filaments. Centrioles, paired cylindrical bodies, each composed of 9 triplets of microtubules, organize a microtubules network during mitosis cell division to form the spindles and esters from the bases of cilia and flagella. Inclusions include stored nutrients such as lipid droplets and glycogen granules, protein crystals, pigment granules, storage for nutrients, waste and cell product. Cellular extensions. The major cellular extensions are cilia, flagellum and microvilli. Cilia, short cell surface projections, each cilium composed of 9 pairs of microtubules surrounding a central pair. Coordinated movement creates an unidirectional current that propels. Flagellum, like a cilium but longer. Only example in humans is the sperm tail propels the cell. Microvilli, tubules extensions of the plasma membrane, contains a bundle of actin filaments, increase surface area for absorption. Nucleus. The nucleus is a spherical or oval-shaped structure that usually is the most prominent features of a cell. Surrounded by the nucleus envelope, contains fluid nucleoplasm, nuclei and chromatin. Control center of the cell, responsible for transmitting genetic information and providing the instructions for protein synthesis. Structure of the nuclei. The nucleus envelope, nucleoplasm, chromatin, the nucleolus. Nucleus envelope. The nucleus envelope is a double-layered membrane perforated with pores which control the flow of material going in and out of the nucleus. The outer layer is connected to the endoplasmic reticulum, communicating with the cytoplasm of the cell. The exchange of the large molecules, protein and RNA between the nucleus and cytoplasm happens here. Nucleoplasm, a jelly-like made mostly of the water, matrix with the nucleus. All the other material flowed inside, help the nucleus keep its shape and serves as the medium for the transportation of important molecules within the nucleus. Chromatin appears as a fine, unevenly strand network, but special techniques reveal it as a system of bumpy threads weaving through the nucleoplasm. Chromatin is composed of approximately 30% DNA, deoxyribonucleic acid, are genetic material, 60% globular stored proteins which package and regulate the DNA, 10% RNA chains newly formed or forming. Chromosomes contain DNA in a condensed form attached to a histone protein Chromatin is comprised of DNA. There are two types based on functions. Heterochromatin, highly condensed, transcriptionally inactive, mostly located adjacent to the nuclear membrane. Eurochromatin, delicate, less condensed organization of chromatin located in a transcribing cells. Function: the nucleus is often compared to the command center as it controls all functions of the cell. It is important in regulating the actions of the cell. It plays an important part in creating the cell's protein. It is involved in important processes dealing with DNA and other genetic molecules. DNA or deoxyribonucleic acid contains the information needed for the creation of proteins, which include enzymes and hormones, and is stored in the nucleus, as already said, in the form of chromatin or chromosomes. The nucleus is the site of DNA duplication, which is needed for cell division, mitosis, and organism reproduction and growth. DNA double helix. RNA are made from the DNA templates. Message RNA directs the synthesis of protein. Ribosomal RNA joins with ribosomal protein to make ribosomes. Transfer RNA binds to an amino acid and hold it in place on a ribosome until it is incorporated into protein during translation. Protein and cell regulation. The nucleus oversees cell functions and regulatory mechanism for keeping the cell healthy and alive. The nucleus controls growth of the cell through the synthesis of structure, protein, energy, and nutrient metabolism. The nucleus regulates the secretion of ribosomes, which are made in the nucleus and are the site of gene transcription. Okay, thank you. अगर आपको मेरा क्लास अच्छा लगे तो आप प्लीज़ लाइक, शेयर एंड सब्सक्राइब कीजिएगा.